So welcome class to next session of today. Today is Friday the 21st and I fortunately I gave you today link on time. It was not that late. I came early from my cycling, but when I was now I'm traveling, it was not like earlier. So I go from 5 p.m. to 7 p.m. Just towards India Gate or somewhere randomly, or to Gurdwara or to some mandirs, and just making my spots. But the situation is so bad at the moment because the level of the poverty that I was able to see. I want to capture it, but it will not make sense to capture the things that to show the people that what is happening. But it's really, I can't describe in words how things are us going through with the one who are living under poverty line. Um, yeah, they were just cooking raw rice on the street with the heat of plastic bags, and the whole family was just having the boiled rice. And I don't know, I saw many instances on the street, uh, mostly looking for some money help, monetary help, and yeah, it was very heartful to see things at this time of our life. So, nevertheless, let's continue with our sessions as usual. We are doing that is about yeah, specimen. We are going very slow, students. Today is 21st May. I think we need to finish by 4th of June, and by that speed that we are going, we have 2 weeks. Maybe I will take your all Saturdays or something. I have to take. Otherwise, this will go forever. And I'm teaching you very slow, going with lots of discussion. But it's good that we are having these discussions and going in detail about each topics today. So, okay, let's start with specimen preparation. That is your light microscopy, scanning electron microscopy, and transmission electron microscopy. Like how we prepare specimens, respect to them. Just a second, मम्मी ठंडा ही दे दो ठंडा ग्लास दूध बादाम पा दो हाँ बादाम कुट के that's it थोड़ा little. So these are the general schematics for the preparation for the specimens for the light microscopy. So on the left is the live mounts, like the one which are alive, and the right one are the one which are preserved, which are fixed. They are not alive anymore. So for the live mounts, we see them. We do the vital stains, and those with vital stains, we check our samples. On the other side, it's known as histology. That's a different department. In the histology, we fix our samples with some reagent, and then we remove all the water, dehydrate them, infiltrate and embed it in a box or in some kind of socket. Then we section it, and then we mount, then stain it, and then we view it. So these are 7 fixed steps that are the one that we use is for the light microscopy. So in order to see the live mounts, we see the organism or tissue cells as close to the natural state because cells are alive, and what we see is quite natural thing. And we do not stain with anything, so that if we stain them, then it will be not the live moment that we are collecting. So that's why, but it has some limited points that the unstained specimens have very low contrast. We can't differentiate between your different compartments within your cell. Whereas the supravital stains may be applied to provide more contrast to identify certain compartments, and these are stains that are not harmful to the living cells. So on the live mounts, I can show you that how a live mount exactly the video looks like, the cells. So this work was done on the cardiomyocytes, neonatal rat cardiomyocytes in PhD. So we were extracting every week on Tuesday, cutting the head, dissect the heads of neonatal rats, 3-day born rats. They were just not even, eyes were not even opening. So we used to get around 60 or 70, and we need to cut their their head, and then afterwards we dissect them from their chest. And after cutting this from the Caesar from the diaphragm, we went onto the heart, and with the tweezers we just get their pumping heart out of it, and we put this pumping heart into the cold buffer, in the cold buffer where the cells remain inactive, and then the primary extraction of neonatal red cardiomyocytes protocol starts going on under the cell culture hood. So that work we used to do 8 in the morning. We used to get our rats 7, and then the operation starts at 8 with them, and it feels horrible if somebody is this pile of Hanumanji, and then they have to cut every Tuesday innocent newly born rats for the sake of experiments. It's horrible. So, but I did that for almost 2 years every Tuesday, and yeah. So this is, I got the results. Okay, never mind. This. ah. So this one is the control cells. You can see how the cells, there are some debris also on the top. They are of not any use, but please check the cells of the different shapes, these elongated cells. And when you see them in the microscope, and they are beating like our heart. So in our heart, we having the sound lubbed up, and they are pumping in the same way. So this was experiments I did in order to find out what is the effect of hydrogen peroxide on these on the hydrogen peroxide. Ay, ay, ay. My mouse is not working today for some strange reason. Yeah, now it's working. So this was the control. Then 100 micrometer of H2O2, micromolar, not meter, micromolar of H2O2 has been added. Then 1000 micromolar, and this is at 10,000. All cells start to die. All the nucleus, they start to pump out. Cells start to sucked out, and nothing was there. So that was the things that we saw under different conditions. And with the help of MATLAB, we calculated the mean contraction area, how much the cells are being contracted, and we played her in the graph. So just showing you. So here we can't see any contrast actually. We can't see cells exactly what is the cell proper morphology. We can't see the internal cells, but still we are able to analyze things. Then these are the fixed. Then now for the histology, for study of the tissues, to the histological studies, the pathology department used to study this a lot. So in this case, we have the specimens. We preserve them with a chemical such as formalin, acetic acid, ethanol, and methanol, and fixations immobilizes molecules such as proteins and lipids. Then fixed specimens are dehydrated by serial transfer through an ascending alcohol series. So like 40%, 50, 60, 70, 80, 90, 100. Then the specimens are infiltrated with melted paraffin substitute or plastic, and placed in the mold to return. Then in the fourth step, after inflating our sample into the paraffin mold, it's time to cut them into thick sections using steel knife or razor on instrument called microtome. So this is the microtome, which is cutting automatically your sections of your specimens. Maybe we can check this microtome. At the bottom, we put a knife, a blade every time, a new blade, and then we lock it. And here, what you're seeing is you put your sample in the paraffin wax. Your samples are being fixed inside them, and you will see that this part will be moving up and down. Or I don't know how this machine will work. I haven't seen this video before. But it will cut the very thin sections. It will come on this plate, and the water will be flowing on this thing, so that the cutting of your slide of your section should be fine, and we can have multiple sections within few minutes. Let's see now. Oh, this is very bad method, not the good one. So in this, it was all manual. There was no water flow coming up. But anyhow, just have a look sometime.
That the tissue is exposed from the wax and can be soaked in cold water for 1 to 24 hours.
This is better. I can share this one with all of you. So in our time, it's not coming up here. Is my voice not coming? Is my voice coming to all of you? Students, please confirm if you can hear me, what I was saying so far. Okay. So the microtomy, that what we see here, is not the good one here. But recently in the market, we are having really good microtome, and they're automatic. We don't have to do it with our hands. This machine go up and down, and there's a continuous flow of water coming up here. So which makes sections coming automatically, and there's a reservoir at the bottom of to collect your samples, and it's a warm one so that the samples could be the results that we are doing, we can get it done. This is a very old one showing here just for the sake. Then after our slides are done, we got the sections, we mount them on the slides, then we stain to achieve contrast or identify. So we do the staining like immunohistogram staining, and then view them under the microscope. So many variations in techniques are used to prepare specimens for light microscopy. Some omits the dehydration, infiltration, embedding, and section steps, and use aqueous staining systems for viewing the whole mounts. Then freezing is also used instead of chemicals to fix the tissues, to examine them quickly and have the components damaged by the chemicals. Like frozen biopsies, tissues in which heat liable structures are to be stained. So these frozen specimens are checked with the help of cryotome. It's not the microtome, cryotome we use, which is encased in the freezing chamber. So we got the permanent slides made from the paraffin sections, not from the frozen sections in that case. So this was all the things that we have seen here. And all these schemes was for the for your live microscopy, light microscopy. Now let's check for the scanning electron microscopy. Of this, I'm not much aware because I never done them practically by my own hand. But I want to show you some results from the light microscopy. This is mass spectrometry, structural biology, summary, lots of Western blots, mRNA expression. Yeah, this one I want to show you. So just look at this second figure. So these figures are from the light microscopy. So I have my neonatal rat cardiomyocytes. In the first row, they are transfected with the control siRNA. In the second case, I transfected them with the siRNA, CTGF. Yeah, negative tissue growth factor. There is siRNA. That is, it's stop the production, the mRNA production of CTGF. Because of that, we see a knockdown in the CTGF production. We see less CTGF in our samples. No. And this CTGF is mainly present at this membrane, cell membrane of your cells, cardiomyocytes. So I dyed, I stained my cells with DAPI dye, ATF6 dye, CTGF dye. So 3 dyes. I have added 3 different antibodies and dye with them, and I saw them under the microscope under different fluorescence. So what I saw was that in the normal case, the ATF6 is also present outside in the cell, outside the membrane, and also the CTGF is present quite nicely outside, inside and very prominently. But when I did the knockdown, CTGF expression got less. That's good. My experiment works. But what I saw that the ATF6, which was present outside, it goes into the nucleus. It was not anymore present outside. So that's the property of ATF6 protein, that whenever there's ER stress, yeah, whenever there's endoplasmic reticulum stress, ER stress is there, so that our ATF6 tend to go from ER lumen to the Golgi. From ER, from the outside, it goes inside to the Golgi, and from Golgi, it goes to the nucleus, the cleave part. So that's proof there is a ER stress signaling is working. Yeah, so that's one example of the images from the cell. I don't have the confocal images. I was not allowed to work. I wish I could show you from my perspective. This work also was published in the scientific reports in the Nature, um, under the heading of reduced activity of PP1 under redox stress conditions in consequence of glutathione mediated transient disulfide formation. This is some extra data, but we don't need them. So for the scanning electron microscope, we fix our samples, again dehydrate. So if you remember in the last time I told you about stubs, yeah, we will see how the stubs looks are. We mount them on the stubs, then sputter coat, we coat them with gold or any palladium, and then we observe. So for fixation, we use glutaraldehyde, paraformaldehyde, osmium tetroxide. And for dehydration is accomplished by carrying the specimens through an ascending alcohol series to 100% alcohol, then to an organic solvent such as acetone, propylene oxide. Specimens for SCM may also be processed in critical point drying apparatus. So specimens then are mounted on the aluminium stubs using the sticky tape. So this is how the stubs looks like. This is one of the stub, like your nail with a round on the top. So applying specimens to stubs arranged in a holder. So there's a holder. We put our these nice stubs there, and on the top we put our sample. And with the sputter coat, we coat our specimens with either gold, palladium, or rhodium in a special chamber to cover the specimens with 10 to 20 nanometer thick metal layer. Then it goes for observation under the SAM, and we can see the things over the videos. So this is the electron column where the electron has been pushed over your sample specimens. Here we put our sample. The machine looks so big, and we can see our display of scanned images on this computer. Then this is TEM. It's not the same, a bit different. So also we fix our samples, dehydrate, infiltrate them, then section them, and apply sections to grid, stain them, and then we observe the results. So fixation specimens are fixed in this TM also with the glutaraldehyde, paraformaldehyde, and then osmium tetroxide. Then dehydration is done using the ascending series of alcohol. Then they are put into the organic solvent such as acetone or propyl oxide. Then they are inflated with the epoxy or plastic resins placed in the plastic molds to harden. So these are the molds. We put our specimens into them. Then these are some like into this plastic. We harden them with make them like a strong one. The specimens by adding some samples like epoxy or plastic resins. Then when they're hard, so we use ultra microtome. It's even higher version of your microtome to section the specimens. So you can see this water. It's cutting this specimens in epoxy, and this round one. This is a very ultra close up of your microtome specimen holder, and this is 3mm diamond knife that we are using here, and resins blocking containing specimens. Knife edge is there, and ultra thin sections of approx 1mm wide. We are getting these sections of our sample. Then these are sections to a tiny metal grid. This tiny metal grid for the sport grid and forceps. So we're carrying it. So heavy metal stains such as uranyl acetate and lead citrate are applied to make certain structures electron dense. So this is the grids are then inserted into the TM and observed. So we put our grid with our specimen on top, and from top this electron column. This is a specimen insertion from here, and it's even complex than your SCM actually, and because we see 3D images in them, and we see our samples on this computer. So We are done with microscopy today, finally. Not this one. I think I shared with you the normal file, right? Yeah. Microscope basically shared. Now it's the advanced version. We have discussed light microscopy and electron microscopy in detail. Okay. Now let's go to the next part, cell culture. Anyone has any questions? Sir, will all these protocols vary with the type of sample used besides the type of microscopy? Yes, yes. I was working with liver, liver sample, heart sample. What else I used? Kidney of mouse for the microscopy that has to go for confocal microscopy. So I used to do sections for them. So for each organ, it was very for the histology part. It was a different way to do it, and also the microtome works differently. The blade was different, and the fixation thing was different because each organ works differently in the different aspects. And if you are looking for what do you say, some fluorescence experiments to check some internal cell organ like mitochondria or Golgi with respect to subsignaling mechanism, whether it's getting high or low. In that sense, you can create your own experiments. So initially, when you will be doing experiments, it's very hard to get the results because we are doing the for first time. But you need to redo it, create 7 to 8 different kinds of variables, so that at the end you are able to get the results quickly. So let's start with animal cell culture. Some brief history. So from 1885, rocks maintained embryonic chick cells live in saline solution for a short length of time, and Alexis carry cultured connective tissues and showed heart muscles tissues contractility over 2 to 3 months. Then in 43, all et al. Produces continuous rat cell line. Then in 1962, Bionicis et al. Published methods for maintaining differentiated cells. Then in 1970s, Gordon Sato they published specific growth factors media required for the many cell types. In 1979, Bordstein and Sato defined the serum-free media, and in 1980 to till now, tissue cultures become an experimental research field and more widely accepted research tool. So some applications of cell culture technique. So it has quite varied rules, very various ways it could be used. So some study of cell behavior without the variations that occur in animals. So artificial organ development, in vitro propagations of virus, vaccine development, drug effect, recombinant DNA technology. Then characteristics of where cells can be maintained over the several generations, leading to the good reproducibility between experiments. And cultures can be exposed to reagents, radiochemicals, or drugs to define concentrations. So finally, it avoids the legal, moral, ethical problems of animal experimentation. Then limitations of cell culture. We have to develop standardized techniques in order to maintain healthy, reproducible cells for experiments. We take time to learn aseptic technique. So everything, whenever you are entering the lab, make sure we are wearing a lab coat. Our our hairs are not open, and our shoes are cleaned also. And after entering the lab, make sure we clean our hands with 70% ethanol. And after cleaning our hands, we put gloves, fresh gloves. Then again, we clean our hands with 70% ethanol, leading to, everything is aseptic then from our body side. And then when we're going to start working on the bench or in cell culture or laminar air hood, make sure your space at the bottom is also 70% with clean with the 70% ethanol. You make a very fresh zap on it, clean it with the tissue paper, then you are good to go. Because aseptic technique is really must. If it is somewhere some contamination is there, whole cell culture is wasted. We need to maintain the cost of media and maintenance of cell line also. So some terminology in the cell culture, that is um, organ culture. So there are 3D cultures, undisegregated tissues retaining some of the features of the tissues in vivo. So we are having organ cultures of a heart, we are having organ cultures of liver, we are having organ cultures of pancreas. So we grow them with the stem cells properly in the same shape. After they get the acquired shape, they start to work function in the artificial conditions properly the way it should be. And the fight is going on with the help of tissue engineering to create the whole organ just with the help of stem cells, and then we can do the transplantations of organs with the help of that. But still, so there's a in my previous lab where I was doing my PhD, so they were working on this organ culture. Yeah, this one. Scientific American. So they have created these organs. Cardiomyocyte, stem cells being
For more than a
pumping.
decade, biologists have been able to turn stem cells into beating heart cells in a petri dish. And bioengineers have had some luck getting stem cells to grow on scaffolds to make simple, hollow organs like tracheas and bladders. But just how hard would it be to make a large, complex organ, like a heart, from scratch? Getting beating clumps of cells to organize into a three-dimensional heart requires scaffolding. At the Massachusetts General Hospital in Boston, Harold Ott and his team start with the scaffold that nature provides. They're using a technique called decellularization, to strip away all the living cells from a heart. The owner of this specimen died only a few days ago. It will sit in this specialized vessel for about a week, while chemicals wash away its DNA, lipids, soluble proteins, sugars, and all other cellular material.
The building, if you wanted to renovate it or repair it, you'd go in, you'd knock down drywall or get rid of cabinets and stuff like that. And basically we're going into the matrix and doing that. By perfusing solutions and detergents, we're getting rid of all the cells.
Afterwards, all that's left is a pale, white matrix of collagen and other structural proteins that once held everything together. Stripping away the donor cells removes the antigens that can cause the body's immune system to reject the new organ. The scaffold can then be repopulated with healthy new cells that are immunologically matched to the patient. The team hopes to one day completely reconstitute working hearts from a scaffold like this. But today, they're focused on how they might develop parts of the heart using the same process. They've taken a valve and part of the aorta from a decellularized heart and sewn it onto a cadaveric heart.
So this part here is the aortic root, including the aortic valve and the ascending aorta. And this has been decellularized, so this is a decellularized graft sewn onto a cadaveric human heart. And you can see here the right coronary artery button and the left coronary artery button sewn onto that decellularized graft. And the same thing here, this is the pulmonary artery and the right ventricular outflow tract here of the cadaveric heart. And this is the graft, the decellularized graft that has been sewn onto the heart.
Amazing. Harold and his team want to check if their graft would be able to do what these structures do in real life: prevent fluids from flowing back into the heart, and feed oxygenated blood from the aorta to the arteries that feed hungry heart muscles. They use a CT scanner at the hospital next door to take a look at their work. Harold pumps contrast fluid through the aorta to check for backflow, and to see if the arteries that feed the heart muscle leak. The team knew an emergency department case could come through at any time, so they had to act quickly.
Actually, sort of, because you could do saline. Okay.
Back in the control room, Harold runs through his handiwork with radiologist Brian Goshadra.
What we have here is a CT of this explanted heart, and you very nicely injected the aortic root. We can see that you've sewn in your syringe to this area. Just to orient us, I'm really quickly going to put it into a 3D mode down here, and then window away so we can see what's left. And let's just blow that up. So
When you look at the sinuses of this aortic root, does that look physiologic to you? What does the anatomy look like?
it looks pretty close to what I would expect in a physiologic human. In this, you can see it's very nice and smooth. So what matters most clinically is the inner luminal smoothness, and really does look pretty good.
The process of decellularization and stem cell seeding could ultimately develop better replacement parts for the heart. These parts might prove more able to repair themselves and could theoretically grow with a young patient. To Harold, this technique could transform medicine.
Last night we were treating a patient who came in and developed sudden onset heart failure and is now currently on a machine that supports her heart and lungs. So seeing these patients and seeing that clinical need and these patients' families and understanding where all this research funnels into ultimately, which is helping patients and developing tools to improve lives of these patients in end organ failure, that's really what gets us going, I think, and what keeps us going.
So we are on the path to do so. So it's actually very old story of 2013, but at the moment I have seen it actually in 2017 from this organ culture creating heart to be pumped fully, to be working effectively. but it's not. It was not big heart. It was a very sample version. It was shown. But let's see. Then there's a cell culture that we have seen or already earlier, where single cells are growing or organized tissues. They are not any more organized tissues, and they work together in order to find out the cell mechanism or cell morphology, how things are working. Then comes your primary cell culture. So these are the derived from the explant or directly from the animals, only for the finite, they will survive for the finite period of time, and they involves all the enzymatic mechanical disruption of the tissue and some selection steps to isolate the cells of interest from heterogeneous population. And some safety level, substances hazardous to health, a substance that can cause cancer, carcinogen, substance that can cause damage to the developing fetus, teratogen, a substance that can cause mutations in the genetic material, can be passed to the next generation mutagen. Then gentamicin, thapsigargin, possible mutagens and some tetrogens possible in the lab. Hydromycin, possible carcinogens are streptomycins. So all these things could be possible in the lab. So be careful when we are working in the lab. Then use of cell culture areas. So cell culture areas, as any of the laboratories, is working area. So do not bring your friends within you. Do not eat, drink, or smoke in these areas, and do not use a mobile phone. So do wear a lab coat at all times, whether in cell culture area or laboratory. Do wear your disposable gloves, but make sure to dispose of them in correct way before you leave the area. And do not wear disposable gloves in the corridors or write-up areas. So basic equipments that we get in the lab while working in the animal cell culture lab, that is horizontal laminar flow cabinets. So they provide more sterile environment for the cells, but offer no protection of the operator. Filtered air enters to the back of the cabinet, is directed to the front and directly at the operator. And the most sterile part of the cabinet is at the back. That is your centrifuges. And we use 100 G is hard enough to sediment cells. So we use higher G forces, which could also damage cells. So we need to be careful about the speed also. So from contamination of the cell culture, we need to be taken care of, like microorganisms who are growing 10 to 15 times faster than the mammalian cells. So like they grow in 8 to 16 hours suddenly, and they are more tolerant to hard temperature, pH, or nutrient supply than the normal cells. So they just need a space and they will start growing. And our cells are most vulnerable, most you can say easy to assess for contaminations whenever we have any aseptic conditions. So this development, so this can lead to the development of antibiotic resistance microorganisms. So mycoplasma is one of the important cell culture contamination. Then isolations of cell lines for in vitro culture, that is resected tissues, cell or tissue culture in vitro, primary culture, secondary culture, cell line, single cell isolation, clonal cell line, successive subculture, immortalization is done, then transformed cell line could go into the immortalized cell line. Some types of cell culture in vitro. So primary culture, they are derived from the animal tissues, embryo or adult, normal or neoplastic, cultured either as a tissue explants or single cell. So initially heterogeneous, become overpopulated with fibroblast. Then finite span of in vitro, they could grow and exhibit all the inhibition part also. In secondary culture, they are derived from the primary culture, isolated by selections or cloning, becoming a more homogeneous cell populations, finite lifespan in vitro, and retain differentiated phenotype, mainly encouraged dependent, and exhibit contact inhibition. Then comes continuous culture. They are derived from primary or secondary culture. And then the immortalized ones are the spontaneously or by transformation vectors. So serially propagated in culture, showing an increased growth rate. And homogeneous cell population has been taken care. And the loss of anchorage dependency and contact inhibition. And infinite lifespan in vitro and differentiated phenotypes. And genetically, they are unstable. So some cell morphology varying depend upon these cell types. So fibroblast cells look these thin, tiny sheets like this, whereas epithelials like together a sheet of cells. And endothelial, they are somehow what we saw in the cardiomyocytes coming together with nice shapes, making a network of cells. And the neuronal cells are like traffic is going on, and they are shining when you see under the microscope, and they make really good network. So we can see all these cells having different um, morphology, different structure. Enzymatically they are working different, mechanically they are working different, morphologically they are looking different. That's why each organ, whether it's a fibroblast or it's epithelial or endothelial or neuronal, um, the basic foundation is different. That's why the organ is different. That's why the tissue that is being made out of them is different. It's like if some house is made from the red bricks, the house will be colored as a red brick all in all. If the house is made from the white brick, different shape, it will be belong to that. So same thing happens in this while working on the cell culture. So we have discussed till now various types of culture. That's a summary for today. We have discussed about primary and continuous. Primary are the one with the normal cell culture without any change in the division rate. In continuous, we have discussed two, finite and indefinite. And the finite one, single cell types, roughly 3 times of divisions, 30 times of divisions, enhanced by the growth factors. With the indefinite, it's nearly the same as finite, but the cells can divide indefinitely. So we also call them cell line. So we keep growing them. So primary culture, according to the cultivation period, they are derived from the excised normal animal tissues, where the continuous cultures, they comprise of the single cell types. That is normal diploid and established cell line. So normal ones, they maintain the degree of differentiations, finite life, and established cell lines, they include tumor cells, transform cells, aneuploid a number of chromosomes, and limitless availability. So we are done for today, I think. We have discussed about all the cell culture today, various types of cell culture. We have discussed about organ culture, normal primary cell culture, secondary cell culture, continuous cell culture. What are the conditions need to be made? What should be taken care? What kind of contaminations could happen? So we have discussed quite basics of today, the animal cell culture, while working in the lab. So tomorrow we will continue from slide 17th about stem cells, how the stem cells are working, and further we will understand what are the various mediums that are required, what does the ingredients of the medium while growing a cell culture, and why is it so important. And further we will discuss the cryopreservation, how you count the cells, so and how the kinetics of your cell culture is working. So all these things is for tomorrow. So that's it for today. Thank you very much. Then we see us tomorrow is not a holiday. We will see us tomorrow again at the same time, and yeah, same place. So we will continue for Saturday because we had a off. Okay, class. Take care. Bye bye.